So good morning everyone again and welcome to the next talk webinar today, Novogene Bioinformatics Analysis Report Walkthrough in mRNA Sequencing. Thank you very much for taking the time to join us today, and we are excited to have you with us. We also hope that this session will give you a better understanding on how to interpret and make the most of your mRNA sequencing analysis result. So I am Menti and I will be the moderator for today's next talk session. Before we begin, here are a few things to know. The webinar will run for about 40 to 50 minutes, followed by a Q&A session. If you have any questions during the session, please feel free to drop them in the chat box and we will address them accordingly. Lastly, to help ensure a smooth session experience for everyone, please keep your microphone muted throughout the webinar. Now it's my pleasure to introduce our speaker, Ms. Jiaxun. Jiaxun has more than 10 years of experience in the field of bioinformatics and have extensive experience in bioinformatics data analysis and also the interpretation. Over the years, she has conducted numerous webinars, seminars, and workshops, sharing her practical insights and bioinformatics analysis with researchers from different backgrounds. Now, we are very happy to have her with us today. And with that, I won't take up any more of your time. Jiaxun, thank you for joining us, and the floor is yours now.
Hi, thank you, Menti. So let me try to pull out my screen. Okay, so my screen is up. So today is a free and easy session. Yeah. And where shall I start from here? Okay. So today we'll be going through the bioinformatics analysis for mRNA sequencing offered by the Novogene. So this is kind of a sessions to go through what the users will have when conducting a mRNA sequencing project. So this session, we will be going through some quick intros on the transcriptomic solutions that are available in Novogene. And then Novogene basically is NextGen's partner. So if you have any transcriptomic related analysis or sequencing required, feel free to reach out to us. So here I'm going to introduce some sequencing approaches or the applications that are available here. And then what kind of, how we handle the projects, and then what are the files or what are the deliverables will be received at the end of the project. And then we will going through a demo reports to show you what are the analysis that will be carried out for the mRNA sequencing, and then how whether it kind of achieve the project objective. And then in addition to the normal analysis, because for RNA sequencing, normally what we are looking at is the expression profiles of our samples. So in addition to the expression profiles, what are the information that we can get from the mRNA sequencing and etc. So at the end, I will also share a little bit on the platforms that is used for project reanalysis and customized analysis. So this is a platform offered by Novogene, and users will be able to use this platform to perform very customized, very personalized analysis for the project. So for transcriptomic sequencing-based solutions, at the moment, I think there are quite a lot of options available. So the most typical one will be the mRNA sequencing. So this one is the captures of RNAs. Basically, it's the messenger RNAs. So we want to see what are the genes that are expressed and most likely translated into proteins to become the functional units. In addition to that, some of the project we might also interested in looking into the non-coding RNAs. So those genes that are transcribed but not translated into proteins. So in that case, we might opt for the total RNA sequencing or the whole transcriptomic sequencing. In addition to that, sometimes we might be more interested in look into the full-length transcripts. So we want to see starting from the 5' untranslated regions, and then the operating frames all the way to the 3' and also the poly A tail. then we might be looking into the isoform sequencing. So this is the full-length isoform sequencing, and in addition to that, we also have the small RNA sequencing, circular RNA sequencing, and then if let's say our samples is a mix of different organisms, then we might be looking into the meta-transcriptomic sequencing. So, and then for bacteria-wise, of course, we are able to do the RNA sequencing as well. It's just that the analysis that will be carried out would be slightly different compared to the eukaryotics, because bacteria normally one gene, one transcripts, while for eukaryotes, normally it will be one gene, multiple isoforms or multiple possible transcripts, because eukaryotes, we do have alternative splicing ongoing. So after the gene is transcribed into the pre-mRNA, so the one that before that can be processed to become proteins, that will be the pre-mRNA, and then undergo the alternative splicings. It could be orders into different types of transcripts, and then at the end, that might be translated into different proteins. So application of RNA sequencing, normally we want to see what are the transcripts that are expressed in the samples. And importantly, we want to see what are the genes that are differentially expressed in different sample conditions. And then in addition to that, for some kind of for widely studied organisms like human or the model organisms like mouse, most of the time we already know what are the genes available and then what are the transcripts that will be produced, but sometimes we might still be able to detect some novel transcripts, so some isoforms that we have not kind of identified before. Maybe it's only present in certain conditions or under certain stress, then we might be also able to kind of identify some new transcripts due to the alternative splicings, and then some organisms that has been kind of less studied. We might be even identified novel genes. That is the genes that have not been discovered before. And then other than that, we might also study the drug resistance across different time course of gene expression, because transcriptome is a screenshot of the transcript at one particular point of time. So the transcriptome or the RNA that we extract in the morning might be different from the RNA populations that we extract in the evening. So before a certain treatment or after a certain treatment, the transcripts might be responsive to the conditions that we are studying. So normally we will have multiple conditions, and then we can study the developments in terms of their expression profiles. And then of course, this will be helpful for us to identify useful biomarkers, and then combining with other omics data through multi-omics, perhaps epigenetics or kind of the genome or the genetics. Basically, we might be able to discover even more informations through the multi-omics strategy. So, and then with all this, we are able to look into the clinical insights. So yeah, so a lot of basically RNA-seq is quite useful in terms of many different conditions. So whenever we want to study the gene expression under different conditions to see what are the genes that are responsive to certain condition, then RNA-seq will be helpful. So to conduct a RNA-seq projects, normally over here, what we are going to see or kind of expect in a project will be starting from the sample preparations. And then for RNA-seq, of course, we normally start from the materials of total RNAs. So here are some of the examples of the sample requirements in terms of kind of to submit for RNA sequencing. So depends on whether we are looking into the eukaryotic mRNAs, or sometimes we might be even look for the strand-specific RNA sequencing. So because we know the genome is double-stranded, we have forward strand and reverse strand, and then normally the gene is present in either the forward strand or the reverse strand. And then in some cases, we are interested in looking into the antisense expression. So we want to see the expression of the genes. We want to see also whether the antisense of it is having certain genes or any regulatory elements that will be handled by the antisense strand. So in that case, the strand-specific RNA-seq will be helpful. So preparation of the samples normally will be at the user sites. So once the extraction is done and then the RNA pass the sample requirements, then we can submit for sequencing. So this one, normally we also need to submit the samples together with a manifest, just to tell the sequencing lab how many samples have been submitted, and then what are the QC that we are tested in the local labs, and kind of informations that are related to the samples, so that when the sequencing lab receives the samples, they know which samples to proceed, and then what are the applications that they need to prepare for the samples. And then once the sample is QC at the sequencing lab, they will issue a sample QC report to tell from their QC how's the condition of the samples, whether they are suitable to proceed for library preparation and sequencing. And then from there, users can decide whether to proceed or to perhaps resend the samples in case some of the sample is kind of off freeze, and then they suggest to maybe try out another set of samples. Then we might be resending the samples until all the samples pass the QC for library preparation and sequencing. And then once we are into the process of library preparation and proceed for sequencing, if the project comes with the BI service, the bioinformatics analysis service, then users might receive a form to fill out. So this is called the BI form. This one is to inform the sequencing lab or the lab handler the data, how many samples will be included in the analysis, and then what are the sample groups involved. So let me quickly show you an example of the BI form. So this is normally how it looks like. So it starts with some general information of the projects involved, and then what kind of analysis is required for this project, how many samples or how much throughput per samples. And then for user who prefer to have both the raw data and clean data, they can ask the sequencing lab to prepare both sets of raw and clean data, or else by default, I think they might only provide you with either the raw data or the clean data. So in addition to that, we need to tell the lab what are the samples names, and then what is our preferred name for the samples, and then whether certain samples, for example, like the A, B, C belongs to the same group, and all the B samples belong to the same groups. And then for RNA-seq, they can accept an alternative groupings. So for example, like let's say I have A groups and B groups. In addition to that, actually A and B is the same sample, it's just before and after the treatments. Then we can also tell us, this is sample 1, sample 2, sample 3, in addition to the sample group A, group B. So in addition to that, we might also need to provide the lab with the genomes, reference genomes that we want to use. If let's say this is a reference-based project, so they will try to match the data or map the data against the reference genome provided, and then from there, the expression profiles will be conducted. And some other details might be also required to help perform the analysis. So some examples would be here. So for example, like this case, it has 6 samples, 6, yeah, and then all of them from humans. And then preferred name in the reports will be this one. And then 2 of them belongs to the wild type, and then another 2 is the KOG, and then another 2 is KOH groups. So, and then provided with the reference genome. And then what are the comparisons required? So we want to compare the KOG groups against the wild type, and then the KOH groups against the wild type. And then what are the Venn diagrams required? So we want to see what are the overlapping parts between these 3 samples, and then perhaps these 2 sample groups, and etc. So example of the plots are also provided in the example sheet of the BI form. So you can always refer to the examples over here. And then some certain species, some additional analysis can be carried out. And then we just need to tell what kind of reference species to use in order to do the analysis. So this is example of the BI form. So once the BI form submitted and then they have done the sequencing, then they will proceed to do the bioinformatics analysis for the project. If this is a project comes with Bioinformatics Analysis Service. So next, just to show you quickly on how the project deliverables will be looking like. So for projects that come without the bioinformatics service, or you might be seeing the tag of WOBI without bioinfo. So users will receive the sample QC report once the labs have done the QC for the samples. And then once the analysis, the sequencing is done, then our users will be receiving the data QC report together with the sequencing data. So depends on users' preference, whether they want to have the raw data or the clean data or both sets of raw and clean data. And then for projects that come with the bioinformatics service or the tag WBI, user will receive the same set of data like the sample QC, the sequencing data, raw and clean. In addition to that, the data QC will comes with the analysis report. So in the analysis report, it will list out all the files provided. So all the analysis result files and data will be provided fully. So how the users is going to receive the files and data. So during the delivery, Novogene will provide a platforms for users to download the data. So users will receive a batch ID together with the password to log into the platform. And then once login is going to be like the normal platform download. So we download the files, and then normally the files, because there are a lot, so Novogene has helped to compress or zip everything into one file for users to download easily. So we don't need to download multiple files or multiple folders. So, but because of that, after the download is done, user will need to unzip the results folders, like using the software WinRAR or 7-zip in order to look into the data and reports inside. So this is how the deliverables is going to pass to the users. So next, let's take a look at how what kind of analysis is included in the BI and how the analysis reports looks like. So let me quickly take out the reports. Yeah. So for this RNA-seq demo report, I only have the PDF versions. So supposedly users will receive the report in HTML format, which can be opened in any web browser. And then the report is going to be interactive. So when there are tables, when there are different plots, users will be able to point and click to switch to different plots inside the reports. But this is a PDF, so it's going to be not interactive, just to show you how the reports or what are the contents is included in the reports. So for RNA-seq analysis report, normally starting it will include some basic information on the project. So what is the reference genome and versions used, and then the time, dates, and also the ID. So in case we need to check with Novogenes on some details for the project. And next, it will include some introductions for like what are included in the RNA-seq project. So this one will have the details on how the library construction and sequencing is done, the workflows included, and etc. So for analysis, I normally will be referring to this particular plots on looking into what are the analysis involved in the project. So for typical eukaryotic mRNA sequencing analysis report, the raw data will go through the data QC before it's mapped to the reference genome selected. So the QC will include the error rate distribution, GC content distribution, and if let's say there's any low quality data, it will also be filtered at this step. So next, after map to the reference genome, this is a very important step because from the alignments, we are going to identify any novel transcripts, and together with the known transcript of the reference genome, we will proceed to do the gene expression analysis. So this is the most important part, also a very important part for the RNA sequencing projects. So we want to see what are the genes that are expressed and then how is their expression levels. So from here, correlation analysis and gene arrangement analysis will be conducted. And then next will be the differential expression analysis. So for projects involve multiple samples or multiple sample groups, of course, we want to see what are the genes that are differentially expressed in certain conditions. So this is where we can find the results. So this particular differential expression analysis will tell us what are the genes that are upregulated, downregulated, or no change constants across different conditions. So normally after this step, we will have a list of genes that are differentially expressed in the conditions that we are studying. So sometimes the list might be long, so a few hundred genes that are differentially expressed. So when we want to look into all these 100 genes, or perhaps even 1,000 genes, what are the functions involved or what are the pathways involved? Normally, we will be conducting some analysis such as the enrichment analysis. So this enrichment analysis, including the GO enrichment and KAGG enrichment, will quickly help us summarize what are the functions that are enriched in the differentially expressed genes. So we can quickly tell, okay, there are maybe 100 genes that are differentially expressed, and then many of these genes is involved in certain pathways or certain functions. Then we will try to see whether this pathway or functions is related to the conditions that we are studying. In addition to that, because we have the sequence data, so it's kind of makes sense for us to also to study the structural profiles of them. So this structural analysis include the SNPs and indels analysis. So when we compare our samples against the reference, then we will know what are the SNPs and indels that are different from the reference genome. And of course, from there, we can also tell whether there is any SNPs and indels or variants among our samples. And then when we study the isoforms of the transcripts, we will also be able to tell us whether there's any alternative splicing happenings, and then whether different samples is expressing different types of isoform from the same gene, or from the kind of different transcripts from the same gene. So this is a quick overview of the analysis included. And in this report, it includes the summary of all the analysis that have been carried out. But because this is only reports, so the full data or full results normally comes together with this report, but it's going to be in the separate files. So the tables that we are going to see in this report will not be the complete tables. Like I will show you the examples later. So this is kind of the QC. So starting from the QC of the data, from here we can see whether certain samples is having high rate of errors, and then whether there's any samples showing some abnormal profiles, and then whether the GC percent is of what we expect for the organisms, and etc. So after the data QC is done, the next step is the mapping. So the mapping, in addition to tell us, sorry, I think the next one. This one is summarizing how much data we receive before and after the filtering or the QC steps. So normally we will retain, I think, at least 90% of the data, I think. And then with the sequencing data quality at the moment, I think sometimes we are seeing 99% of the data being retained. So yeah, next, the alignment. So this one is the step we align the clean data against the reference genome selected for the projects. From here, we can see how much data is mappable to the reference genome, and then this number can directly tell us whether the reference genome is suitable for the project or suitable for the samples. So this one, because sometimes some samples, they might not have very good reference genome, or the reference genome might not be very close to the samples. In that case, we might seeing the percentage of total map will be low. So in that case, users can consider also the de novo approach. If let's say there's no good reference around for the samples. So, but if let's say we are seeing more than 90% or even more than 80%, most of the time is going to be okay. And then it will also show how much data is mapped to the exon regions, or basically the gene regions, how much is mapped to the intronic regions, and then how much is mapped to the intergenic regions. So yeah, quick summary. So if we want to really look into the alignments of it, normally we will need to look into the data files provided. And because the data files provided is going to be in certain format like the BAM format, so here in the report, they also suggest users to download a software called the IGV if, let's say, they are interested in looking in the alignment details. So how each of the data, each of the sequencing reads is mapped to the reference genome. So this is the screenshot of IGV with the alignment files. And then for RNA-seq, this is how the kind of the alignments looks like in the IGV, because eukaryotic genes, normally we have alternative splicing happening. So we are going to have exons and introns for one genes, and then the mapping will show which exons is kind of having high mapping rate, which exon is having low mapping rate. And then this is only a screenshot, example screenshot of the IGV. So in all the RNA-seq report, no matter which project you are looking at, you might see the same pictures, but this is just to show you the example. So your data, normally we need to download the software and open the alignments to really look into that. So next step will be after we do the alignments, then the pipelines will try to identify any novel genes or any novel transcripts. So this one, they will try to see whether there's any regions inside the reference genomes not annotated with any genes but is expressed. Then they will try to see whether that particular region is actually having a genes that have not been discovered before. So all these genes that is novel, not in the annotations, they will be included in the total gene sets to proceed for the next step, gene expression. So gene expression is to tell for each of the samples, for each of the genes, what is the number of sequencing data observed. So normally we will have expression tables in this format. So the full data will be inside the files provided. So normally it's going to be a table file, which can be opened in spreadsheet application like Microsoft Excel. Each of the rows normally is one gene, and then each columns represent one samples. So we can see what are the genes that are highly expressed across all the sample or in certain samples, and then what are the samples that are having high expression for certain genes. So it's a big tables. And then normally Novogene will provide multiple tables of this, including the raw sequencing count, and then normalized sequencing count, and also the normalized and kind of balance across all the samples. So there might be multiple tables in this. So it depends on user, they are looking into the raw sequencing count or looking into the normalized data. And then further look into the gene expression levels. They plot the expression level across the samples for kind of in the box plot in this case. So this one can quickly tell us whether all the samples are having all the kind of number of genes or ratio of genes that are expressed, and then whether there's any obvious outliers if, let's say, we have a lot of samples. And then from the correlation analysis, comparing the expression profile across different samples, we can also quickly tell which sample is close to which samples, and then whether the sample replicates are close to one to another. So this one, if we have a lot of samples, normally we will try to see whether the samples of the same groups are having the similar patterns of expression, or whether the sample replicates are all close to each other. So if we find certain samples is having expression profile that is very different from other samples, even the replicates from the same groups, then we might be trying to see whether that one belongs to the sample outliers. And another way to look into the comparison or the correlation will be the PCA plot. So this one is even more straightforward to tell us how which sample is closer with which samples. So sometimes the experiment might involve conditions that are not so different in terms of the expression profiles. So perhaps I think one of the projects that I involved before is they are trying to see whether there's any gene expression profile changes with certain supplements. So we don't really see very high differences or very much differences before and after eating the supplement. This is when we look into the overall changes, because if, let's say, after eating the supplement, then the expression profile is changed drastically, then that might actually be something wrong, because supposedly our normal gene expression should functions like before we eating nasi lemak or after eating the nasi lemak will be supposedly not so much different. So this one, if we see, if we do not really see very high difference between sample groups, for example, we don't see the nasi lemak groups before and after separately very significantly, normally that is still okay. Because I think some of the researchers might be concerning my sample groups before and after, they looks very similar. Is this all right? So I think most of the time, if they are similar, they are still okay. And then sometimes the conditions might be very different comparing one to another. Then we normally will be seeing biological replicates being grouped together, and then different sample groups will be forming different sample clusters. So this one will depends on the experiments or the samples that are studied in the projects. And then next is looking into which gene is shared across different samples or different sample groups. So this one depends on the BI form that we fill out. And then the Venn diagram will be compared according to the samples or the sample groups that we have selected. And then next will be the important parts on the kind of help to identify the genes that are differentially expressed. Most of the time for RNA-seq related experiments, this is where the most of the insights that we can discover. So differential expression analysis, there are different approaches to work on this. So for the pipelines, basically we have two main different, I think, normalized methods or different software to do the analysis. So depending whether our experiments involve biological replicates for each conditions or no biological replicates. Most of the time, we actually recommend users to have biological replicates for RNA-seq projects. So one thing, of course, the statistical model will work better for samples involving biological replicates. Another one, if let's say this project involve kind of at the end of the projects, users are aiming to publish the results in kind of submitting to journals, publishing the papers. Most of the time, the reviewers will look into whether the experiments involve biological replicates. If no biological replicates involved in the projects, they might reject the paper quite immediately. Yeah, so that's why if let's say the project is aiming for publication, suggest normally we include perhaps at least 2 biological replicates per samples or 3 biological replicates, so we can have a more robust differential expression analysis and also kind of pass through the reviewers, and then not getting rejected immediately because there are no biological replicates involved. And then depends on different experiments or software used, then they might involve different normalizations methods. And normally the differential expression cutoff will be set by default using this one. So log two-fold change more than 1 or less than -1. So this is show the differences between the changes, how many folds differences are they, and then how statistically significant are they. So normally we'll be using the adjusted p-values less than 0.05. So this one, some users, because their experiments involve samples condition, there are not too much difference. So they might want to adjust the stringency in terms of differentially expressed genes screening. So this one at the end, I will be showing how to do that if let's say users would like to change the different cutoff at this step. So by using the default cutoff, the reports will summarize how many genes are actually identified to be upregulated or downregulated. So this one, again, this is only the screenshot or showing the first few lines of the full table. So normally the full tables is inside the results files provided. And quickly, it will help to summarize comparing, let's say in this case, I have 2 sample conditions, comparing this against this. How many genes is considered upregulated? How many are considered downregulated? In total, how many differentially expressed genes have been identified in these projects based on the default threshold? And then this will be also kind of plot into the bar plot in this case. So if let's say we have multiple differential expression analysis carried out, then we will be seeing comparing, let's say I have treatment 1, treatment 2 compared to the controls, whether the treatment 1 is going to give me more differentially expressed genes or treatment 2 is going to be more drastic in terms of changing the gene expression profile for the samples. And then one of the signature plot for differential expression analysis, this is the Volcano plot. So this one normally it will tell how many of them, kind of what is the magnitude in terms of their differential expression. So going upper parts. So if the plot is very high in the plot, normally that means the significance level is high. So this is the negative log p-values. And then the left and right, it will shows whether the differences is up regulations compared to the control or down regulation compared to the controls. So Volcano plot. And then next is the cluster analysis. So it will also try to look into the gene expression differences in terms of the heat map. So what are the genes that are highly expressed? What are the genes that are lowly expressed across different genes? So yeah, this one is a quick summary of how difference with comparing different sample conditions. So sometimes we might be filtered the genes before we plot this, or else actually most of the genes will be showing no difference across different conditions. So in order to highlight the genes that are differentially expressed, normally this one will only involve the differentially expressed genes. So after we have identified the genes that are differentially expressed, next we want to identify what are the functions that are involved. So whether this gene is affecting certain functions or certain pathways is affected by the sample conditions. So normal methods to do the analysis will be the enrichment test or enrichment analysis. So this one is to try to check how many genes, let's say it's involves certain functions over here. So how many genes is involved in that functions from the total differential expressed genes obtained. So let's say we have 10 genes that are differentially expressed. 3 of them is involved in certain process, perhaps glycolysis. Then whether this is significant, we are not sure. So we need to compare to the total background genes or total genes that we are studying. So if let's say out of the 10,000 genes that we study, and then 3,000 of them involved in glycolysis, then this 3 is not so significant. But out of the 10,000 genes that we study, only perhaps a very small percentage involved in glycolysis. But in our differential expression genes, we are seeing a lot of genes involving in glycolysis. Then this is called an enrichment. So we can tell glycolysis is something involved in the conditions. So based on these principles, the analysis can be carried out for different functional annotation terms. So one of the famous terms famous functional profiles that is used is the gene ontology or the GO. So this one can quickly tell what are the GO terms that are enriched. So for example, like in this case, oxidation reduction process, and then the gene ratio is 12 out of the 94 differentially expressed genes compared to the background. So out of the 1,752 genes studied, 61 actually involved in this process, but we are seeing 12 genes in the 94 differentially expressed genes. So the p-values, sorry, this one has been cut off. This one, the p-value is 5.5 e negative something. So if let's say this p-value is less than 0.05, most of the time we can tell this particular function is significantly enriched in the differential expression. So we might further study what are the genes involved in this process, and then how is this process associated with the experiments or the conditions that we are studying. So, and then it will also help to plot the significance level in the plot like this, and kind of show the results, summarize the result in different types of visualizations plots. And then in addition to the GO, so this is actually a different level of GOs to show which part of the GO graph is enriched in the projects. And then in addition to GO, I think a lot of people will also look into the KEGG enrichment, because GO tells basically the process involved biological processes, cellular components, and molecular functions. And then some people will be more interested in looking which pathways is involved or which pathway is affected by the conditions. So this one, based on the KEGG database, the enrichment will tell or will help to highlight which pathways is significantly enriched in our differential expression analysis. So again, based on the p-values, like in this case, glycolysis, having the p-values 3.8, e negative 0.6 is going to be considered significant, enriched in the conditions that we are studying. So again, the kind of showing the results in different plots. So normally this plot could be included in the papers if, let's say, this project is going to be published. And then which kind of in the report, they will have to highlight which pathway is considered the most and which one most affected by the sample conditions. And then again, full results will be inside the tables provided in the results folder. And lastly will be the protein-protein interaction networks. So after identifying the genes that are upregulated, downregulated in the projects, then we might also be interested in looking into whether certain genes of them are having interactions based on papers or based on publications. So this one is based on the database, the string database. If you try to see what are the genes or their coding proteins interacting with each other or having certain correlation with one another. So this one can help to quickly identify whether certain gene is actually the drivers for all the expressions. So if, let's say, we are having one gene and then they are related to many other genes, and then all of them are actually also differentially expressed, then we might be kind of interested in looking into this one, because it could be the one that driving all the changes inside the samples. So this protein-protein interaction, full results provided, but normally they will suggest to use certain software in order to look into the graph. So normally I think the Cytoscape software could be useful in terms of looking into the protein-protein interactions results. So the full results provided in files like this. And then in addition to the differential expression or the expressions study, and then if, let's say, certain users they are also interested in looking into the structural differences, so alternative splicing analysis can help to identify whether certain genes is spliced into different transcript, and ultimately will be having different sets of proteins, and whether these different sets of proteins are expressed in different sample conditions. So they, in addition to identify the differential express genes, they also will help to identify the differentially expressed isoforms. So this sometimes could be interesting because from the same gene, different isoform could be different regulatory impact on the samples. So perhaps certain isoforms will help to trigger certain pathways, while the other isoform from the same genes might be triggering a totally different functions or totally different impact on the samples. So normally, this one I will be kind of interesting to look at, but normally this one will be put as secondary, because short read sequencing, sometimes we are just guessing how many exons are there, how the exons are connected to one another, and then only then we try to compare different isoforms. Most of the time, the gene expression will be still the focus of RNA-seq projects. So in addition to the alternative splicing events, sometimes this one will be interesting as well if, let's say, our samples is of different individuals. But of course, if, let's say, the sample is from the, let's say, cell lines where we do not really expect them to have different SNPs and indels inside the samples, then this might be not interesting. But if let's say the sample is collected from different individuals, in addition to looking into the genes differences across different conditions, we might also be interested in to look into whether there's any polymorphisms carried by all these samples. So SNPs and indels from the alignment, it will try to identify the differences across different samples. So, and then SNPs or the variance annotation will be carried out, including like whether the variance is inside the exon regions, intronic regions, and etc. Most of the time, we don't really capture or not able to capture the intergenic parts because RNA-seq ultimately is to study the genes. So sometimes if we have some expression at the intergenic regions, so this might not be very significant. Most of the time, we are looking into whether certain genes is carrying certain variants. If let's say we are looking into RNA-seq data. So at the end of the reports, include the software that is used together with the version, and then the parameters that is used. So this one will be useful also for whoever would like to publish the projects into paper or perhaps to write the thesis. And then actually for the project, they will also provide a methodology write-up. So in case you need some help in putting all this information into the write-up for papers, and they actually have provided PDF versions of the methods write-up. And some helps in terms of how to handle the zip file if let's say the file received is zip. So most of the time, Windows, they suggest to use the WinRAR or 7-zip, while for Linux, basically you can use the built-in commands to do the uncompression. And then the data might involve many different file formats as well. I think most of the time, like the tables, pictures is not a problem for most of the users, but some formats is actually quite specific. Let's say like this VCF is for variance data, BAM is for the alignment files, and then normally we need to have certain methods or certain software to help us handle this kind of file formats. And then at the end, the reference that is used for the projects. And before I end the demos here, just want to show you if let's say the projects involve human or mouse or any medical species. In addition to the analysis that we are seeing, it actually also will include the analysis on fusion genes, oncology genes analysis, and then reactome, and then the disease ontologies, and etc. So all this is specific to human and mouse samples because the database basically is only on human and mouse. So if we have a different species, a plant species, or perhaps like the one just now, I think is the amoeba or something, then we might not be able to analyze all this analysis. So that's why this only applicable for medical species. So if you have medical species, your report might be longer than the one that we see just now. And then for other eukaryotic RNA-seq, normally that is the one that we are seeing. So I think we are reaching almost the end. So before I end the session, just also want to show you that to answer the question just now, if let's say we have different thresholds that we are looking at, so different differential expression analysis cutoff, we want to change the log2 fold change cutoff, or we want to change the p-value cutoff, or perhaps even we want to restructure the whole analysis, what we can do is using the platform provided by Novogene. So this is called the Novomagic. For the projects kind of work with Novogene, they can help to set up this platform for users to handle the whole project or the analysis. So for projects, let's say we identify certain outliers, unfortunately for the projects, we want to exclude the samples from the analysis. We can do it over here, like identify the samples that we want to exclude, and then from the grouping, we regroup the samples. After we have worked on all this, then we can actually go to the analysis part, redo everything using the new set of samples or new set of selected groups. Alternatively, we can also run the analysis specific one by one. So for example, like let's say I just want to change my differential expression cutoff, so I can do it over here, like looking into the DGE list, and then change the different grouping scheme. If let's say I have regrouped the samples, and then different cutoff, whether I want to use adjusted p-values or just the p-values, and then what is the cutoff that I want to use. Perhaps I want to increase it to 0.1, and then the cutoff I want to reduce to none, or even increase it. So after we have adjusted the cutoff that we want, we can proceed to redo the analysis and then check the analysis whether it's done over here. So when it's done, then we can look into the result. And then we can also download the reanalysis results. So yeah, I think this is supposedly to be the one. In case of, oh sorry, it's still running. So this is the one. So it's still running. So we can always go back to the project analysis over here. Oh, sorry, the project task over here to look at the analysis that we have carried out, and then download the results. Or in the projects, after we review everything, we can also come up with a new reports that we like the one that we see just now, but with the updated results. And then other than that, the Novogene also have the toolkits for the users to kind of fully customize the analysis. For example, like plot different things. So this one, if let's say users download the data and then filter the data manually and then want to plot certain plots for the papers, they can make use of the tools provided over here. And then Novogene also come with several AI agents. So for users need some help in terms of preparing the data, or use some general English to instruct the AI to do some plotting, some agents are going to be helpful over here, and even do some paper polish. Yeah, so that is the Novogene platform. So summarize everything. So just now we have gone through the available sequencing approaches or the solutions provided by Novogene for transcriptomics. And then we also have looked into how the projects will be conducted, and then how the analysis will be carried out, what are the analysis is included in the projects, and what are the supports that users will be receiving after the projects. So with that, thank you everyone for staying with me in these sessions. So yeah, pass back to you, Mickey.
Yeah, thank you, Jasmine. Thank you for your insightful bioinformatics walkthrough. So now we will open the floor to the audience for the question. Does anyone have any questions to Jasmine? You can feel free to unmute yourself and ask, or you can type your question in the chat box. So let's wait for about a couple of seconds. Yeah, I think there's no question from the floor. So before we wrap up, we would love to hear your thoughts about today's session. If you have a quick moment, please fill out the survey form that I pasted in the chat box. So your feedback is very valuable to us, and we will share the e-certificate once it's ready. Also, if you have any question after today's next talk, you can feel free to contact us through sales@nextgen.my. Once again, thank you, Jashin, and also all of you for joining us today. So we truly appreciate your time, and we look forward to see you again in our future events. And thank you, everyone. Have a great day ahead. Bye-bye.
Thank you. Bye.